Now, I want to share you some stuff about the ramp utilizing receptors. And one of the things about that multispan is really good at is developing cell lines that potentially contain complicated signaling pathways, or complicated receptor and receptor partners, basically. So ramp requiring receptors are, ramps are modifying proteins, they're 1 through 3. They determine the selectivity of some receptors like the calcitonin receptor and the calcitonin-like receptor for different ligands. So essentially you have 2 receptors, you have 4 different ligands, you have calcitonin, CGRP, amylin, and adrenomedulin. And with these combinations of things, so the ramps and then the different ligands, you wind up with 7 pharmacologically different receptors. So one of the challenges with this assay is to develop assays for these receptors in cell lines that don't have ramps so that you can add the ramp of choice. So you can add ramp 1, ramp 2, ramp 3, so that you can identify the pharmacology that you're interested in. And so it actually, everything kind of makes sense. So we went into the literature and pulled out data for this ramp for CGRP. And in the data, in the literature, the background cell was HEC293 cell. And so we used the HEC293 cell for this assay. And in this, we actually found that when we had an empty vector, we still got signaling with CGRP. So we got signalings with CGRP. When we added the ramps, we got more signaling, but you know, the issues are that these particular cells, the HEC293 cells, actually had signaling in the absence of adding the calcitonin. So this is the empty vector. Empty vector had signaling. The other vector had signaling when you added the ramps, but there really was, the cell basically had a high background. That's the bottom line. So the literature said that HEC293 cells were a good cell for this particular platform. Problem is that we found that in this, we wound up with a background, and so these cells were not appropriate. So we wound up looking at CHOK1 cells. So here's CHOK1 cells, ramp with the empty vector. There's actually no signaling at all. This is with calcitonin. This is with calcitonin-like receptor. But when we added the ramps, now we started to get signaling. So through this, we were able to switch to the CHOK1 cells and develop assays and cell lines for all of the different 7 distinct pharmacologies by calcitonin-like receptor and calcitonin. There's another complicating thing in this, though, is that increases in receptor expression actually overcomes the ramp absence. And so here, this is the clone, one clone with very little expression of the receptor. Here's the receptor expression. You can see as you increase receptor expression, now you start getting an increase in signal. So you have to pay attention to a couple things. You have to pay attention to whether the cells have endogenous ramps, probably quite a bit of expression of ramps, and you also have to pay attention to how much receptor expression you have in those cells so that the endogenous ramps don't play a role in actually the pharmacology of that receptor. So we learned a lot with this, and through this, we were actually able to develop, like I said, the cell lines. So here's the amylin 1, 2, and 3 receptor. Here's the FACS analysis for that. And here, basically, everything is a CGRP stimulation. This is with the ramp 1, which is amylin, and you can see we got nice signal here. This is with the ramp 2. So this is amylin 2, and this is amylin 3. So you get different pharmacologies, 1, 2, and 3 receptor with ramp 1, 2, or 3. This is CGRP and adrenomedulin receptor. And again, this is a calcitonin-like receptor expression. And this is the ramp expression shown in those cells. And from this, essentially, this is the CGRP and adrenomedulin. So ramp 1 for CGRP, ramp 2 for adrenomedulin 1, and ramp 3 for 3. And here's the empty vector again with no signal. So essentially, we've developed the calcitonin receptor family of receptors. Here's all the different names of those receptors, the receptor, our catalog stable cell line, and then the endogenous ligands for that. Now, one thing that we could do if we were asked is that it would be very interesting to look at all the different ligands with respect to the differentiation of these receptors and how the different ligands signal, but we have not done that yet.